هنحاول we'll try to speak for 3 minutes. احنا متأخرين يعني هنتكلم بسرعة مش هتأخر. وأول start and then we'll handle to Dr. لطفي جعفر and then Dr. Lawal and then Dr. رؤوف اللهوي and then حضرتك at the end. حضرتك الآخر. عشان نحترم الجائزة بس ولا حاجة؟ عشان الجوائز. بس اوكي خلاص. ماشي حسن. Do you want us to speak at the podium? ممكن speak at the podium أحسن. Yes. بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله. My name is Hassan Azazi. I'm a professor at the American University in Cairo, and I'm the chair also of the conference and the president of the Egyptian National Committee for Biochemistry and Molecular Biology. And I would like to welcome you to the 18th International Conference of Biochemistry and Molecular Biology. At the beginning of my welcome remarks, I would like you to stand up for 1 minute for the victims of the Medicaine as well in Libya, as well as the earthquake in Morocco. Thank you. Thank you. It is again my great pleasure to welcome you to the 18th International Conference of Biochemistry and Molecular Biology, organized by the Egyptian National Committee for Biochemistry and Molecular Biology of the Academy of Scientific Research and Technology. The theme of this conference is Towards Better Future for Humanity, and it was carefully selected to reflect the major contribution of biochemists as well as molecular biologists to the betterment of humanity and life on Earth. We have a very extensive program with 38 speakers as well as 40 poster presentations that from different countries, including speakers from Egypt, Germany, India, Saudi Arabia, South Africa, Emirates, and the US. They will discuss cutting-edge research in different areas. We have several sessions, including biomedicine, nanomedicine, neuroscience research, genome research, and bio and chemi informatics. I would like to highlight the 40 poster presenters. Most of them represent young researchers in the field of biochemistry and molecular biology, and this should be highly celebrated. And this poster session will be during the lunch break, which will be from 1:45 or now from 1:30 to 2:45. We have one speaker that is unable to make it, and we have used the 15 minutes to expand the lunch break and poster presentation instead of 1 hour to 1 hour and 15 minutes. Poster presentation will be judged by 10 judges, each assigned 10 posters for selection of awards. This year we are going to offer 4 Best Presentation Award for young researcher. Again, without ranking them in first, second, and third, each one of them will be Best Poster Presentation, and each of them will receive a £3,000 cash award. I would like to recognize my dear colleagues and friends, Professor Dr. Raouf Al-Allawi, former president of the Egyptian National Committee of the Biochemistry and Molecular Biology. I would also like to recognize my dear colleague, Professor Dr. Lutfi Jafar, Dean of College of Science and Engineering at the American University in Cairo. I would also like to recognize Professor Lawal Bilbis, President of the African Federation of Societies of Biochemistry and Molecular Biology. We were supposed to have also Professor Dr. Iqbal Parker, which is the Secretary of the International Union of Biochemistry and Molecular Biology, but unfortunately he was hospitalized yesterday and was unable to make it to Cairo. I would like to recognize members of the scientific and organizing committee of the Egyptian Committee of National Committee of Biochemistry and Molecular Biology, who have contributed tremendously to the organization of this conference. They have put countless hours and made a significant contribution to the organization of this conference. I would like to also to recognize members of the Logistics Committee who have helped in the registration and other preparation for this conference. Finally, I would like to recognize you as participant in this conference and encourage you to make connections, make new friends, make plans for the future to advance your research and so on, and have, of course, a productive and exciting time. I have one announcement that for certificates for this conference, all of them will be sent by email within 2 to 3 weeks after the end of this conference. And I just like to remind you at the end of my concluding remarks that tomorrow, inshallah, second day of the conference will be via Zoom or a webinar. And again, thank you very much for your presence and kind attention. Professor Dr. Lotfi Ghaffar, Dean of School of Sciences and Engineering at the American University in Cairo.
Good morning and welcome to the 18th International Conference on Biochemistry and Molecular Biology. On behalf of Dr. Ihab Abdurrahman, the Provost of the American University in Cairo, it gives me great pleasure to welcome you to Egypt, to the American University in Cairo, and to the School of Sciences and Engineering. Dr. Ihab Abdurrahman couldn't be here because of a prior commitment, but he conveys his greetings and best wishes for the conference and all participants. At the start, I would like to extend a special welcome to our distinguished speakers, guests, and attendees, especially Dr. Gina El-Fi'i, Vice President of the Academy of Scientific Research and Technology, who is here on behalf of Dr. Mahmoud Saar, the President of the Academy of Scientific Research and Technology. I also would like to welcome Dr. Lawal Bilbis, the President of the African Federation of Biochemistry and Molecular Biology Societies, and of course, our own distinguished university professor, Dr. Hassan Azazi, the Chairman of the conference. Luckily for you, I am an engineer, so I wouldn't be able to talk anything technical about this conference, so you are going to be spared a very long speech. However, I know the little I know about the two disciplines is that they really get or try to understand the basic level of life. What is life at the very foundational level, with implications, as I understand, on medicine, agriculture, the environment, climate change, and many other fields. So it's a very significant field. I am lucky at AUC and in my school to have colleagues in the Department of Chemistry, Department of Biology, Institute of Global Health and Human Ecology, and even the Center, Magdi Yaoub Center for Heart Diseases. So we do have many units that I could always go back and get small education on this topic. So I encourage you and invite you to please interact with our colleagues from these various units during your stay. I also invite you to please take a tour of the campus. We are very proud of our campus, and I think you will find unique facilities. So please do that. With this, I just wish you a very pleasant and productive conference and the best of luck. Thank you.
Thank you very much, Professor Dr. Lotfi Ghaffar. It is my pleasure to introduce Professor Lawal Bilbis, President of the African Federation of Societies of Biochemistry and Molecular Biology. Professor.
Good morning, everyone. My name is, as Professor Hassan said, Lawal Bilbis. I'm a biochemist and also Vice Chancellor of Usman Danfodiyo University, Sokoto, Nigeria, and the President of Federation of Societies of Biochemistry and Molecular Biology. This is a platform that brings all biochemists and molecular biologists in Africa under one umbrella. On behalf of the society, I wish you success in the 18th International Conference on Molecular Biology being organized by the Egyptian Society of Biochemistry and Molecular Biology. My friend, Professor Parker, who is the Secretary General of the International Union of Biochemistry and Molecular Biology, could not be with us today. He was to leave South Africa yesterday. Unfortunately, he took ill and he was hospitalized. He sent his goodwill, a personal goodwill, and also of that of International Union of Biochemistry and Molecular Biology. Egyptian Society of Biochemistry and Molecular Biology used to play a central role in the study and the experimentation of biochemistry in Africa. Unfortunately, for a couple of years, the Egyptian Society of Biochemistry and Molecular Biology has not been playing the role it's supposed to play in Africa. I want to use my presence here to also have a meeting with the society to see how we can bring Egypt back to where it belongs, in the center stage, in the study of biochemistry and molecular biology in Africa. The society brings all biochemists in Africa and molecular biologists and interact with the other fellows of biochemistry and molecular biology across the globe to promote life and well-being of humanity as the theme of this conference, towards a better life for the humanity. May God guide and protect us. May He also see us through this. Our prayers are with our brothers in Morocco and Libya. May God see them through this trial. Amen. Thank you and wish you all the best.
Thank you very much, Professor Lawal Bilbis. I think because of time, we are going to hurry up and present the awards. The first award goes to the Professor Iqbal Parker, and as the Secretary General of the International Union of Biochemistry and Molecular Biology, he has done a great effort to communicate with the Egyptian National Committee of Biochemistry and Molecular Biology. It is unfortunate that he is not here because he has been hospitalized, but we'll offer the award to the Professor Lawal Bilbis. Professor Lawal. The second award goes to Professor Lawal Bilbis himself, who is the president of the African Federation of Society of Biochemistry and Molecular Biology. Professor Lawal. The third award is to Professor Dr. Mahmoud Saar, President of the Academy of Scientific Research and Technology, and he has been a strong supporter of the Egyptian National Committee of Biochemistry and Molecular Biology over the years, and it is my pleasure to offer this award on his behalf to Professor Ahmed Salim. The fourth award goes to Professor Dr. Ihab Abdurrahman, Provost of the American University in Cairo. For those of you who may not know, Professor Abdurrahman has been a strong supporter of the Egyptian Committee of National Committee of Biochemistry and Molecular Biology. Actually, the last three conferences, the 16th, 17th, and this year the 18th, were hosted at the American University in Cairo, and again, due to strong support of Dr. Ihab to this committee. So this award will be offered to Dr. Professor Lutfi Gafar on his behalf.
And finally, and many thanks for Professor Dr. Hassan Azazi for his unbelievable efforts for this conference, and he is the chairman also of the Committee of Biochemistry and Molecular Biology in the Egyptian Academy of Science. Thank you, Dr. Hassan.
I think now we will invite our speaker for the first session to start our conference. So if someone can help me to the boards. عبد العزيز فين أو أي حد بصراحة.
بسم الله الرحمن الرحيم. إن شاء الله نبدأ first session. We have Professor Ahmad Sultan, Georgetown University, Washington, D.C., USA, and also Alexandria University, Egypt. Dr. Ahmad.
Bismillah ar-Rahman ar-Rahim. First of all, I would like to thank the committee of the conference for inviting me today, and especially special thanks for Professor Hassan Azazi for his great efforts and support. And today I have 25 minutes to talk, and my title of my talk today is a novel delivery system for targeted bacteria ghosts loaded with epigenetic regulators, nanoemulsion, a potential treatment strategy for breast cancer therapy. So I'm going to, that's my disclosure. The overview will be, number one, the challenges of the approaches with the nano drug delivery. Then, the potential of external physical and disease treatment and the drug delivery. We're going to focus on the bacterial ghost as a new promising platform for drug delivery, targeting tumor cells, and this bacteria promising strategy for treatment of breast cancer, human breast cancer. And finally, shaping the future of drug delivery in cancer treatment. So I am going to take you for a very short journey about what we are doing in my lab in the United States or even Egypt. And our focus will be the shaping the future of drug delivery of cancer treatment, and we keep our eyes on Egyptian vision for 2030. So the challenge that's facing the nano-based drug delivery system for cancer treatment is till now focusing on transporting and maintaining the therapeutic concentrations in the target biological sites. But actually, we should keep our eyes or envision on the future and focus on the following: enhancing target efficiency of the drug, increasing the capacity through specific barriers, and one of the great problem which is long-acting delivery, and improve pharmacokinetics for better personalized medicine. So the main challenges, the nanotechnology for treatment of cancer, now celebrating around 30 years of success. Even so, and with all improvement in cancer therapy, is still facing a lot of challenges, like the clinical problems and translational problems, cost, long-acting drugs, capacity for loading, and all these challenges still keep that even a few hundred of this nanotechnology now reach to the market for treatment of several types of cancer. If we see we have here 3 types of successful types of extracellular vesicle used for nanomedicine. And these 3, including the exosomes, macrovesicle, and apoptotic bodies. Even so, we still have a lot of problem with this. Maybe that's from my experience with the exosome. That's decorated with different types of protein on the cell surface, which can enhance the immune system. Plus, it can be loaded or packed with several anti-cancer drug or molecules like microRNAs, some type of inhibitors of HDAX inhibitor and so on. This external vesicle is very easy to prepare, and we can isolate it from dendritic cells or mesenchymal cells or macrophages, and we can also packed with several types of drug for targeting anti-cancer, targeting cancer cells. But we still have limiting drug loading efficiency and insufficient clinical grade production. And these 2 limits the use of such vesicle to be successful targeting strategy for treat several types of chronic disease, including cancer. So we're still having another hope, which is promising delivery system with bacterial ghost as a new promising platform for drug delivery and molecules. A couple of years, we have a very interesting finding. That's we find that in aerobic bacteria, this type of bacteria living in hypoxic tumor cells itself and in microenvironment around the tumor. One of these is Salmonella typhimurium, which is a lot of bacteria, but we are focusing on Salmonella, and I will tell you why. And this type of interesting type of bacteria, called analytic bacteria, that has a lot of private good potential for detection, for direction of cancer therapy. Not only that, but seem to have unique ability to target cancer cells, especially for those they have resistance against radiation and chemotherapy. So this type of bacteria that's living and the tumor itself can produce several types of molecules like flagellin. This flagellin can bind with type of receptor, toll-like receptor 4 and 5, and enhance the immune system, or activate a strong immune system to attack the cancer cell. Plus CDs, that's a type of molecules that can switch the inactive broad type of drugs to active type as anti-cancer drug. And by this way, we can use this type of bacteria as it is, as a natural, or even after molecular modification. So why we use the bacterial ghost? Bacterial ghost actually, that's empty cellular envelope, but reserving their surface structure, and that's very important, and that can carry around 250 femtoliter per 1 particle. Why we use this type of salmonella particle as a new delivery system for several type of molecules, drug, nano-emulsion drug, and so on? Because number one is still onolytic bacteria living in the environment of a tumor itself or inside the tumor. And this, by its mobility, can dispersion inside the tumor and amplify their function. Not only that, can carry specific genes, even large type of pool of genes, and this can also very easily genetic manipulated. This type of bacteria use active targeting and stimulate immune system. And we found that the microenvironment itself in the tumor is more bacteria friendly. So in this case, we have a lot of types of this type of bacteria we can isolate, genetically modified, and they use it as targeting for cancer therapy, for cancer cells. How that's happened? In case of salmonella, as you see here, these salmonella infected cells, after infection of the cells through the flagellin, that's called pumps, antigen in the surface of this type of bacteria, can activate T3 and toll-free, toll-like receptor 4 and 5 to activate the T cells, cytotoxic T cells, and macrophages, and start to enhance immune system to attack the tumor cells. So, not only that, that's type of bacteria, salmonella specifically, that trigger the infiltration of the immune cells through the CD4 plus, CD8 plus T cells in tumor burden, and enhance the type of this population of cells to attack more and more of the cancer cells and decrease the size of tumor very effectively. So our research here, or strategy will focus, is very interesting to explore the development of newly designed bacterial ghosts. These bacterial ghosts loaded with several, not only one drug, but for first time we saw that several epigenetic regulators in nanoemulsion. That will be a potential target strategy to target several types of cancer, but we succeed firstly with the breast cancer. If we see the approaches for the epigenetic modification, a lot of the DNA methylation, histone methylation, and histone acetylation, all this can activate several character and features against the cancer cell to inhibit cell proliferation and invasion, motility, enhance apoptosis. All that can be modulated using the epigenetic modifications. From this point, let us see the time to plan what we did in this regard. Our strategy has 3 phases, categories. The first category will be that production of bacterial ghost and will characterize what type of bacteria we are using. Second, nano carrier emulsion, not only for one drug. We used several drugs together, a combination of drug, which is new. Then anti-cancer assessment of these new formulated particles. That's type of abbreviation we're going to use. Important MIC, minimum inhibitory concentration, and MGC, maximum gross in concentration. So the bacterial ghost preparation has 3 steps. The first one, determine the MIC, MGC, through several of compounds and chemicals, and then we have optimization method, as we will see with each other, and then full characterization through various methods. So nanoemulsion itself has two types of steps. So the first one, using oil and water emulsion protocol, and then using several determination and characterization method like PDI through morphology changes, and determined by TEM, FTIR, drug entrapment efficiency, drug release assessment. And then we can see, after all that bacteria ghosts are promising strategy for nanodrug delivery and its application with the cancer cells. First one, let us see the bacteria ghost preparation and characterization. We start from heavy growth of these bacteria, uncontrolled, to reach to no growth at all. So we have bacteria lost the growth after we evacuate the bacteria from all cytoplasmic content, DNA and the proteins, and all molecular stuff inside. Then we reach to no growth at all, but we still have the envelope of these bacteria intact. At first, we released the DNA completely, and last portion of DNA removed from these bacteria was after using 60% of ethanol, and that's the last portion of it. And then we released most of the protein content, and last portion also released using ethanol. Then we try to see before and after what's happening. In case of control, you can see several types of the colonies in control itself, and before optimization and remove most of the content of DNA and protein, still we can see some small colonies around. But finally, after the optimization steps, we reach to no growth at all, but still we have the envelope intact, completely intact of the particle of bacterial ghost. And then using a special dye, risperidone, that's blue fluorescence dye, can be changed to the pink color if these cells of bacteria still viable or contain any content of cytoplasmic content. And you see the control here show that's pink color formation, that's mean completely active bacteria, and ghost, prepared ghost, give the blue color, fluorescent blue color, that's mean don't change to pink because of absence of viable colonies. Another tool within blue staining to check the viability. In case of control, we have complete active bacteria with all cytoplasmic content. That's stained in blue color, as you see. Before optimization, we still have some rows of bacteria containing the cytoplasmic content, and they still show up here. And then after complete optimization, we remove complete content of the cytoplasmic compartment of this salmonella, and we have only the envelope intact with all pumps that can enhance immune system. Then we need to prove that the cell membrane of outer wall, cell wall of these bacteria is still in very good shape. So we have the intact, we have negative staining, which stain the background, give the background black, as you see, and under microscope you can see it white road. This white road shows there is no difference between control and empty ghost. That means the cell membrane is okay and intact, and we can use for any packing for any type of anti-cancer drug. After that, we use the safranin staining for cell wall. Safranin give a pink color, as you see, with negative gram bacteria. So if it's give this type of color, that means we still have the negative black bacteria. After evacuation of the cytoplasmic compartment completely of this type of bacteria, we have this road empty, but still having the pink color for the cell membrane. That means we still have this sort of negative gram bacteria. So using TM characterization, you can see the wild type bacteria with this black color with cytoplasmic content, full of all cytoplasmic DNA and proteins inside. Then the second one, after evacuation and expulsion of all the content of cytoplasmic part, we leave transparent compartment with intact cell wall. You can see that's the small pore that we did to remove all, evacuate all cytoplasmic compartment and leave cell wall intact, as you see. So then scanning electron microscopy show that the wild type has complete intact cell wall. The same for bacterial ghost after evacuation of the cytoplasmic compartment, and they have this pore that we already use it to get rid of all content of the cytoplasmic part, and we have intact cell wall. So we can use it one more time for using to packing with several type of molecules, and especially anti-cancer molecules. The phase 2 will be nanoemulsion preparation and characterization. And in this part, we used oil portion. First time we used for one of the trial, 1%. We reached to 66% entrapment efficiency. And then we used the oil portion increased to 10%. We used entrapment efficiency for nanoemulsion drug, which is two types of drug, one anti-cancer drug, the other one HDAC inhibitors. To have synergistic effect for this type of treatment to attack the or targeting cancer cells, we reach to 97% entrapment efficiency. So after that, this emulsion go through several type of the characterization, like average size, average Zeta potential, average PDI, entrapment efficiency, method of emulsification, all this to show that we are on the right track. Then, to ensure the proper loading, we use several methods for characterization, like FTIR, the spectrophotometric analysis, SEM, TEM, CFM, all that to prove that we have a proper loading capacity. So if you see the FTIR spectroscopy, we can see that's free drug, and then drug in nanoemulsion, and the drug after loading with the bacterial ghost. Then we used the spectrophotometric analysis to know that this drug that we use have a fluorescence capacity, so we can detect it when we packed the bacterial ghost with this type of drug. So we find the fluorescence between 420 to 490, and this fluorescence can be helpful to know that we are in the right track, and we actually reach to a proper loading capacity for our carrier. So then under confocal fluorescence microscope, you can see the bacterial ghost after evacuation of the cytoplasmic content contain no color, no fluorescence dye at all, because that's still empty. But after packing with our nanoemulsion drug, which contain anti-cancer drug and HDAC inhibitor, we can see this fluorescence show up suddenly, and then we can detect that we under right track, and we already packed our carrier with the right drug and adjust the concentration of this mixture. Under transmission electron microscopy, we see that nanoemulsion droplets loaded inside the bacteria, as you see, and emulsion. In that case, we reach to successful loading for our particles. Then the last phase will be anti-cancer assessment. To know its work or not, we used actually panel of anti-cancer drug with anti-cancer cell line and the tumors in animal model. We reached to very good result with bacteria as a carrier with breast cancer, second level prostate, last level with liver cancer, or HCC. The free drug itself, we determined IC50 of this drug. It was 112 micromolar, but after we have this free drug switch it to nanoemulsion droplets, we reach to 40 micromolar, which is very good at the decrease IC50 of the drug. And then after that, after loading bacteria with the drug, we reach to IC50 around 50 micromolar. I won't show you that's the difference between the free drug and nanoemulsion, or the particle after loading inside the bacterial ghost. Then panel, we used two types of cells, MCF10A, which is near normal cell breast cells, and MD231, triple negative breast. Actually, we have a panel of the over 12 breast cancer cell line, but actually I won't show that as the comparison between near normal and triple negative cells. The significance difference was in case of MD231, as you see, if compared with control, or even empty vehicle, or free drug itself. On the other hand, with MCF10A, near normal breast cells, the number, actually the breast cells doesn't affect at all with the emulsion, our emulsion, and the progression and the proliferation as a normal for these cells was normally. So in this case, we see that our bacteria targeting cancer cells. Also, we have an animal model, in vivo model, xenograft, and by direct intratraumal injection of our carrier, Salmonella carrier, for this mixture of the drug or emulsion nano drugs, we reached to decrease the size of tumor around 60 or 70%, and this result is still under publication. So that's why I need time to present publicly. So, but it was promising, very promising, that even in vivo and in vitro, our system is still working very well. Colony formation and apoptotic induction, we can see the difference between control and empty vehicle, nano-free nano drug by itself, and the bacterial ghost loaded with the nano emulsion. You see the induction of apoptosis was the maximum in case of the bacterial ghost, since the bacteria targeting cancer cells. But in case of near normal MCF10A, the induction of apoptosis, even in high concentration, was so limited compared to the cancer cell. That's another evidence that we are targeting cancer cell in our system than near normal breast cells. In case of mobility assay, we can see here the control after 0 and then 24 hours. What's happening for the difference in the space of the wound healing assay. Empty vehicle, mostly same like control, and the free drug, little bit decrease, but not much. But the big significance was in case of loading Salmonella particles loaded with the our emulsion, nano emulsion drug. And the difference was around 50% or maybe more inhibition for cell mobility. Also invasion assay, other apoptotic signal, caspase activity, all that's enhanced. Capacity for metastasis also decreased. So we have a bunch of the data support our finding, but because of time, so I just won't share with you still this under press, maybe in the next month or so. But even so, I would like to share with you what's going on in my laboratory. So let us see the future of drug delivery for cancer therapy. We have still a lot of it changes. As I told you that nanomedicine reached to 30 years of research and success since the first introduction. But till now, we have a lot of problems with the preparation of this nanomedicine. As I told you before, that's cost, maybe one of the reasons, how to control the loading capacity, targeting efficiency through some barriers like brain, intestine, and so on. Another thing, how we can focus to find the good way to reach to personalized medicine. So the future that can be shaped, and we can see we have with good technology that we reached now, we can reach to a good maximum capacity using the nano emulsion technology. So merging the advanced technology in drug delivery using artificial intelligence, that will be one of the reasons to know the proper structure to target a specific disease. And then using the macro or micro robots, that will be another way. And I think in Japan, I have a meeting last month, and they already discovered type of the robots that can detect, the small robots, macro robots can detect the tumor itself inside circulation. As you know, any tumor has a fingerprint, secretes some molecules inside the circulation. like circular DNA or something like that, this robot can detect this type of molecules inside the blood and then trap it. And after that, after a specific analysis, you can know what type of tumor and what stage of this tumor. And not only that, all the molecular profiling system of this tumor, so we can have reach to better technology and the treatment strategy. That will enhance the targeting efficiency of the tissues for tissues and cellular level for a specific organ like lung and brain and intestine. And this can help for next generation drug delivery for chronic disease, specific for a long-acting delivery system of our molecules. And in the end, we reach to the specific or better treatment for personalized medicine. That will help for using nanotechnology and nanomedicine targeting specific cancer cells. So the conclusion here, that series of innovations and combined efforts that will help for drug delivery technology with low-cost magnification platforms that will provide significant momentum toward new drug delivery using nanotechnology, nanoemulsion, nanomolecules, or even better, using for exosomes and the other molecules that exist in the market now for delivery, since the loading capacity is still small and also long-acting capacity is still very short. So we are targeting a new biotechnology platform. And our study with all this small amount of data that I share with you today, and you will see the paper next month, with everything, that will be a good proof, highlight the possible therapeutic value of the bacterial ghost as a novel targeted drug delivery system for different chemotherapeutic. Nanoemulsion is specific for the type of cancer that has resistance for radiation and immunotherapy. That will help as anti-cancer effect for several types of cancer molecules. In the end, I would like to thank my colleagues, Professor Huda Mahmoud, for helping with bacteria and for evacuation of this type of molecule, preparation of molecules, and my student, Sarah Hassan. She has actually most of the effort for preparation of nanoemulsion and also preparation of the ghost and packing of ghost, hard worker. That's very important to say, and she is going to join my laboratory in the United States very soon, so we prepare her for that. Dr. Chris Albanese, Director of Imaging Institute, helping us with all the nice images that you will see in the paper next month. And best greeting for my research team in Egypt. I say hi for all of you, and I'm so proud to be with you here as every year. Thank you so much for the organization and the committee of conference. A special thanks for Dr. Hassan Al-Zazi for his help and support. I'm so happy to see all of you. Thank you for your time and attention, and any question?
Thanks, Dr. Ahmed. And I think we will postpone questions to the end of this session. Shukran, Dr. Ahmed. And now we are calling Dr. Amr Amin. Dr. Amr Amin from the United Arab Emirates University, Al Ain, United Arab Emirates. Welcome and hello.
Okay, assalamu alaikum. First of all, I really want to thank the organizing committee for inviting me over here. Despite the fact I've been in this business for maybe 30, 40 years now, I have never been getting this chance basically to talk to people here from Egypt. So really thank you very much for this opportunity. It's been wonderful. Okay, so I am not used to be stuck to a podium, but I will try to manage that. Today I'm going to start to take you to a journey that we've started maybe 30, maybe like 15 years ago. And in order to reach to the point that I will give you the cutting edge data that we have, I really have to take you through the whole history. So bear with me on that. Most of the work has been done in this university, which is UAE University. However, in the past couple of years, I was in University of Chicago, so it's like a merge between these two universities. So I will give credit for everything at the very end. So clearly, I'm going to have a little bit shift from Dr. Ahmed's talk. He was very much focused on breast cancer. In my lab, we are very much focused on liver cancer and to be more general GI cancer, gastrointestinal cancers. And most of the data that you will be looking at here is either liver or colorectal cancer. I still have a couple of graduate students working on pancreatic cancer and gastric cancer as well. But most of the data that you will actually be seeing is liver and colorectal cancer. So again, clearly for a very good reason. I mean, it shows itself whether it's a mortality problem or incidence problem. I mean, it's 25% of all cancers are GI cancer. So it's something that we really have to address one way or another. And this is the saffron that I will try to convince you today that it's good for you. It's extremely good for you. And as you will see from this image here, you guys might have sort of guessed why it's very expensive. It is very expensive because saffron is this little stigma that you see. And these stigmas, in order to actually collect, you have to collect by hand. So imagine if you have hundreds of acres of this flower and you want to collect this, and there is absolutely no way that you can collect it by anything other than hands. And that's why it's extremely expensive. And of course, it's very easy also to falsify, and people sometimes you can get like a gram of saffron for 10 pounds, which is clearly not saffron, because if it's saffron, it's very expensive. It's not going to be that at all. Okay, so of course, saffron is not new. I mean, people have used saffron for a very long time, and you know that it is good for 100 different things. And again, this is taken from this particular article here, and you can see that it's been, explored for different illnesses and lesions and everything, including even brain cancer. So I mean, it's been around, and people knows a lot about it. So my story, at least from my perspective, started, I think, 2009 or 2010, where we start looking at saffron, and we start to extract saffron as a crude extract, and then we see if it has any preventive or protective effect on liver cancer. In this particular case, you can look at here. Of course, I'm not going to bug you with all the details, but you can clearly see that we have HCC group and a bunch of other groups that has been protected with saffron. And if you look at this particular one here, which is where the cancer was developed, you can see clearly here the lesion. And of course, this is labeled by a specific stain. And then in other animal groups where have been protected, you can clearly see that this lesion is almost disappeared. And that was very exciting for us, but was not exciting enough to actually make it to hepatology. And it actually went to hepatology and published there. And then we were even more surprised when we have the cover page or the cover story in Science News. So since then, we thought, well, I mean, why? I mean, we are not the first one to do anything on liver cancer, but it turns out to be, yes, we actually are. And it turns out that one of the problems with liver cancer, I'm very sure this audience know this a lot more than me, that liver cancer is very hard, extremely hard to diagnose. And that is why it's been resisting any kind of treatment, because most probably when people know that they have liver cancer, it's too late. So diagnosis really is a major problem in this. And that's why when we came up with this data, it was very well accepted, and it was all over the place very much. Then we looked at saffron also in preventive capacity in colorectal cancer, and that also was published. And then, as all of you scientists here know, when you publish anything with crude extract, you always get this flag kind of thing, or resistance, if you like, that, well, crude extract has tons of different things. I mean, so which one of these? In saffron, for example, saffron has about 160 biomolecules. 160 biomolecules, and you are saying that, well, the crude extract prevent cancer, that's good, great. But if I want to transform this into medicine, I'm going to have to look at a biomolecule rather than just a crude extract, a bunch of things going together. And that's what we start looking at. We start looking at crocin first, which is one of the bioactive molecules, which actually give the saffron. If you guys use saffron at home, it's a little bit yellowish. This yellow color is crocin very much. And we looked at this is the crocin. By the way, crocin, for the chemistry people, crocin is a carotenoid, which is one of the very few carotenoids that happens to be soluble carotenoid. So it's, again, this is sort of advantage for us. We've done the same thing. We looked at whether crocin can prevent liver cancer, the same setting, and again, you get the same data kind of. If you induce cancer, you get the tumor, and then if you prevent it with crocin, you almost get rid of the tumor. It's all gone. So molecularly, we looked at it, of course, in vitro, and we've seen this is typically what you see in liver cancer. All of these pathways and all of these things are involved. And then once you add crocin, magic happens very much, and you get inhibition of the major pathway that you would be imagined that actually take a part in liver cancer development. For example, inflammation gets, in this case, blocked by crocin, oxidative stress, and you name it. I mean, everything. And again, this was published. And also the molecular mechanism of it was published against liver, colorectal cancer in this case. And then this is brand new stuff that we have published exactly a couple of weeks ago, where we asked this question, whether crocin can treat. Again, I'm very sure that you are aware of this kind of terminology. When I say prevent cancer, I mean that you have, for example, an animal model, and before you develop cancer, you give the rat some sort of crocin or whatever drug that you are using for a couple of weeks, and then you induce cancer and see if this progression of cancer will be slowed down or not based on the fact that they are already taking the drug. That is preventive. Treatment is more exciting, because treatment, you are looking at rats that has been developed or has developed liver cancer, so you can actually see the tumor, and then you give them whatever drug that you want to test or assess. That is therapeutic, and that's what you're going to be looking at right now. So I thought, of course, I have tons of data on this, but I thought this worth, the whole data, basically, because what you're looking at now here is, this is the control, the first one, perfectly happy liver and everything. And these are the HCC where you develop the tumor. You can see here the liver is messed up very much. Here is the news. All of the other ones that you see here, whether it's HCC plus sorafenib, which is a regular drug that people use for treating cancer, liver cancer, or crocin, or a combination of both, all of these guys down here has been started at this level, because this is, again, therapeutic. So all the rats started at this particular shape. So all of them were heavily loaded with liver cancer. But when you treat these mice with sorafenib, which is the typical drug people use, or crocin, which in my case, this is the drug I am interested in, or a combination of both, you can clearly see here the transformation of the liver back to almost normal. And that is very exciting for us. In fact, that was later on patented, and I'm going to show you the US patent after that. Before I get into this, when you deal with cancer in general, there are 3 pathways that you are really interested to see your drug working against or with. Inflammation, you really need to see if your drug can inhibit inflammation, because usually inflammation is associated with development of liver cancer or cancer in general. You also need to see whether you can block proliferation or division of the cell, clearly, because tumor by definition is uncontrollably division of cells. And also you need to see if you can promote apoptosis or cell death or not. So if you can achieve these 3 goals, blocking inflammation, inducing apoptosis, blocking proliferation, then you're good. And this is the first one you can see here. This is, again, we're going to go into the details of this, but these are markers for proliferation. You can clearly see and quantify it here and everything, that crocin, whether it's by itself or in combination with sorafenib, severely block proliferation, which is a good thing. And again, these are a bunch of other markers. Apoptosis, same thing. You look at the quantification here, you can see apoptosis is induced with crocin alone or crocin with sorafenib, which is, again, that's exactly what you want. And of course, the inflammation, NF-kappa-B is one of the key regulators of that, and COX-2 as well. Again, you can clearly see the inflammation here is reduced with all different kind of combinations. And this is the patent I was talking about. And that's why it slows our publication. We published it only a couple of weeks ago because of that. Because when you do anything like has patent or anything, you have a block. You cannot share anything before you get this protected first. And that's why, despite the fact that we have this data maybe like 3-4 years ago, but we could not do anything about it until we get this protected. And this is the publication I'm talking about, which is a couple of weeks ago. And now we're gonna, from now until the end of this talk, we're gonna be talking about the other bioactive molecule, which is safranal. This is the structure of safranal. And again, we looked at safranal for prevention, and we got a patent on that as well. And then we start to look whether safranal can treat. I think now you understand the difference between prevent and treat. So treat means that, again, we're going to have the same setting. You induce cancer, and then after cancer has been induced, you have treatments of a combination of safranal, in this case, and sorafenib, which is the common drug. Again, same thing. All of them, all the livers from here down, started from this. All of them had this structure and this texture of tumor, which is basically developed liver cancer. And if you treat it with sorafenib alone, you still have a little bit of nodules here and there. If you treat it with safranal, you almost have none. You have maybe one here. And then if you treat it with a combination, again, you still have one. So basically, treating with safranal is as good as if you combine it with the actual drug that's being in the market, so, which is, again, a very good thing. This is just how they look in tissue. And again, we are looking here at the capability and the capacity of safranal to actually block the fibrotic capacity of the liver. And again, a couple of markers here is clearly that it actually block the fibrosis. And I'm not going to, again, bug you with all these data, but we looked at bioinformatics, proteomics, and all of this stuff. And this is a summary of all of that, because this shows you that we have a bunch of genes that have been very invested in different pathways. Again, I'm not going to go over all of these pathways, but I want to drive your attention to this because I will come back to this in a few seconds. So just remember that that among these pathways that has been, sort of blocked by safranal was cellular respiration. This will be important in a few minutes. Same thing, safranal does block proliferation. Again, these are the markers, as I mentioned before. And one thing that we have done a little bit further here is to look at how safranal exactly does that. How does it block proliferation? We have published that actually in earlier work that safranal has some sort of effect on blocking the cell cycle regulator, one of the cell cycle regulators. So what you are looking at here is a docking assay where we've seen, or we have shown here, that safranal has a sort of tendency to bind to a protein called CDC25B, which is very much a phosphatase, if you like. I'm very sure some of you are students, and this is actually one of the exceptions I always teach in my class. Phosphatases in general, unlike kinases, they inactivate their substrate by removing phosphate group, except this guy. This guy actually removed the phosphate, and as a result of removing the phosphate, it activates its target. Why I'm saying this? Because now imagine a situation like that. This is how the cyclin binds to CDC25B, and once they do that, the cell progresses from G2 to M and so on and so forth. It turns out the safranal comes here and binds to CDC25B and block its capability to bind to cyclin B, which means that the cell cannot proceed in cycle anymore. And that is a direct evidence of a direct interaction between safranal and a molecule. And this is very important for future, because if you are developing this as a drug, you really want to know how this drug works. And this is how the drug works, at least at this perspective. Again, apoptosis does the same thing. I'm not going to go through the details here, but I want to show you also something that happens to be safranal also block or interact with TDB1. And what is TDB1? I know I'm out of time. I'm going to try to rush a little bit. So I'm not sure if you remember this from cell biology or other molecular courses that you had in early on. Whenever the DNA start to duplicate, they have to do some nicks in the double strand and then unwind, and then they start to do the new DNA. And usually these nicks or these breaks are filled in by topoisomerase 1 and TDB1. So these two components come together and sort of heal this in order to get to a brand new DNA.
Dr. Amr, you have 2 minutes.
2 minutes?
Yes.
Okay. So if you take my word for it, so basically it does, block this interaction, safranal, and that's why you get this. Of course, I'm not going to go over that. Patent, published. Yeah, this is the stuff that I told you. You remember this respiration thing? So it turns out that these data I got only few weeks ago. It turns out that also safranal, this is the metabolomic data that you can see. And again, to cut the whole thing, it turns out if you are familiar with something called Warburg effect, which is the guy who actually discovered this got Nobel maybe 100 years or 90 years ago. And it says that cancer cells depend on very much aerobic glycolysis. And why is that? Because cancer cell wants the cell to make lactate and take the lactate as a building blocks for new cancer cells. So typically all cancer cells, or most cancer cells, have this aerobic glycolysis activated. Why I'm giving you all this data? Because our metabolomics data suggests that these two particular enzymes here, and I'm going to show you again, safranal binds to this enzyme and also binds to this enzyme, these enzymes are blocked by safranal. In other words, cells with safranal treatment, they cannot use Warburg effect, and also they cannot use the regular respiration event. So imagine now the tumor is under the stress that they cannot use aerobic or anaerobic, procedure. So now it's like suffocation, and then they eventually die. Okay, this is what I'm doing now, and since I noticed that all of you are interested in material biology, which is Dr. Ahmed mentioned, so I'm gonna just walk you through this. These data I got a couple of days ago, actually, where we developed MOF, which is a metal organic frameworks. This is a very big thing nowadays, and I'm not gonna go. These are the MOFs that we developed, and these are the MOFs that happen to be loaded with safranal. And biologically, they have antibacterial effect, especially in gram-negative bacteria. And also, most recently, we have shown that MOF with safranal can induce, killing or block the proliferation of cells by maybe tenfold over the MOF itself. I had this activity. Clearly, we will not be able to do that. So these are the people that in the lab and do most of the work. Most of them, actually all of them, are graduated. And these are the collaborators that I have been working with: NYU, Johns Hopkins, Chinese Academy of Science, Zurich, University of Chicago, Berkeley, Yagi group particularly, Howard University. These are the people that I am in collaboration with, and the funding is this. And I'm gonna leave you with this. I'm very sure that you are scientists and you know what this means. So this is your journey when you get to publish any work. So, thank you very much.
Okay, thanks, Dr. Amr, for this.
Sorry.
Thank you.
Again, if you take questions now? Or?
No, after the session, inshallah. Dr. Mohammed Zakaria, please. Next lecture from Professor Dr. Mohammed Zakaria Gad, Vice Dean for Faculty of Postgraduate Students and Scientific Research, and also he is the former Dean of Pharmacy and Faculty of Pharmacy and Biotechnology at the University, at the German University in Cairo. Welcome, Dr. Mohammed.
Assalamu alaikum. معلش, I'm replacing Professor Parker, so I'm supposed to be after the coffee break, but I think Professor Hassan Azazi told me to start now, and this was great pleasure. I'm Mohammed Zakaria Gad. I'm Professor of Biochemistry and Vice Dean of Faculty of Pharmacy, and Faculty actually of Postgraduate Studies Scientific Research at the German University in Cairo, GUC, and former Dean of Faculty of Pharmacy as well. First, of course, I would like to thank Professor Hassan Azazi and my dear colleagues at the Egyptian National Society or Committee of Biochemistry and Molecular Biology, Professor Ahmed Salem and Ahmed Sultan. Also, thank you very much for chairing this session. So let me start the title of this work is Expression of Hydrogen Sulfide Synthesizing Enzyme in Breast Cancer, Influence on Cancer Hallmark. If you know me or have seen my work before, I've been working in the field of gasotransmitters since the 90s of the 20th century, and من التسعينات تقريبا, where nitric oxide at that time was booming. And my first publication was 2000 on the modulation of nitric oxide synthesis inflammation with a dear colleague from Cairo University. At that time, nitric oxide research was accelerating, and the nitric oxide molecule was selected by several researchers to be the molecule of the year 1992. And later on, this field has been extensively extended to cover several research in this area. Nowadays, there are two other gasotransmitters have been added to the family, which is hydrogen sulfide and carbon monoxide. These are the three gasotransmitters that has been known now to be biological mediators in our body, as I'm going to explain. All of them have actually very bad reputation of being very toxic molecules at high concentrations, either from environmental pollutant or so on. But actually, it was surprising at that time to many scientists to find that them are actually very potent and very effective biological mediators in our body. They are all gases, and they are all small molecules. Actually, you can find that they exert physiological function in almost every physiological function in our body, either in the vascular system, in memory, immunity, inflammation, angiogenesis, post-transcription modifications, mitochondrial function, and so on. Actually, almost they have function in every organ we have. And if you go to one of the conferences on gasotransmitter, you can find scientists working in many different areas that cover these very biological active molecules. I'm not going to go extensively toward their functions, but at the molecular levels, of course, nitric oxide was the first molecule that has been discovered in 1987. Later on, carbon monoxide, and then lastly, hydrogen sulfide. They all share some functions. They are freely diffusible. They have anti-inflammatory effect. They are antioxidant, vasodilator, anti-apoptotic, and cardioprotective. Of course, many of the biological studies have been done in the cardiovascular system, but today I'm going on and talk in a different discipline in cancer. Nitric oxide is known to be synthesized in our body by three enzymes: eNOS, iNOS, and neuronal nitric oxide synthesis. I'm not going to go in detail on this. While carbon monoxide is synthesized through heme oxygenases, while the third molecule that I'm going to focus today is hydrogen sulfide. The second mediator is potassium ATP channel, actually. And of course, they have different properties and they have different half-lives and so on. Of course, you can go through literature to find millions of literature now in different directions about these molecules. What I'm going to say today that despite their important physiological functions, they also are involved in many pathophysiological disorders in our body, including cancer. And here is the potential roles of hydrogen sulfide itself in cancer. Of course, you know this hydrogen sulfide, everybody worked in the lab in hydrogen sulfide. It has a very bad odor and well known as a molecule, but actually it has physiological and pathophysiological role in our body. Here in cancer, you can find that hydrogen sulfide is involved in evading apoptosis, in cancer metastasis, in angiogenesis, in cancer metabolism, in tumor growth, and also in DNA damage. So it's involved in by various directions in the oncogenesis or tumor growth. What is really confusing in the studies on literature that usually these molecules either applies for nitric oxide and also for hydrogen sulfide. They also they usually have what is called bell-shaped character. Bell-shaped character that usually at very low concentration and have very high concentration, they are usually cause cell death, as you see here. While on average, at higher concentration, it causes cell proliferation. So it's very concentration dependent, actually, this kind of effects. And this is how to be careful when you work on these molecules. And when we find sometimes is pro-apoptotic, sometimes anti-apoptotic, sometimes induce cancer, sometimes prevent cancer. So it's kind of concentration dependent, and also this is also not applied for the endogenous synthesis of this molecule, but also when you apply it from outside by the exogenous application of, for example, sodium hydrogen sulfide or molecule that produce hydrogen sulfide. So this is kind of tricky, and you have to be known that this is usually have biphasic or even multiphasic properties on our cells. Hydrogen sulfide is synthesized in our body by 3 enzymes well known as CBS, or cystathionine beta synthase, or CBS, CSCE, cystathionine gamma lyase, and the third one is 3-mercaptopyruvate sulfotransferase, which is transaminase. All 3 enzymes actually, the precursor for this molecule is cysteine. They are coming from cysteine, and they have been found in different tissues in our body with different expression, and for example, CSS mainly present in the cardiovascular system, but they have differentially expressed. Nobody knows what is the expression levels in different tissues and why 3 enzymes are present, not 1, no 3 or more, but this is what actually known right now, that these are the enzymes that synthesize H2S in our body. Moving to breast cancer, we have been focusing on, in this study, on the role of hydrogen sulfide metabolism in breast cancer. As you know, breast cancer is not an easy disease. It's not a single actually disease. It has several histological subtyping and also have several molecular subtyping, the luminal A, luminal B, HER2 positive and triple negative breast cancer with different expression of the receptors. For example, triple negative breast cancer, which is the most aggressive one, has no estrogen receptor, progesterone receptor, and epidermal growth receptor. So actually, to go further in this, we focused in our study also in triple negative breast cancer because it is the most aggressive subtype with poorest diagnosis. Shows, we studied actually hydrogen metabolism, H2S metabolism in all cancers, but we focus more on triple negative breast cancer because it's the most aggressive one. It has very poor prognosis. It has larger tumor size correlated to larger tumor size and higher chance of lymph node metastasis. And one of the main causes of metastatic brain tumor, in addition, chemotherapy usually is not very much successful in eradication of this disorder. So now the logical framework, this was the introduction. So what is the logical framework for the study we have done in our lab over the past 2 years? That we studied first the expression of the 3 enzymes in the breast cancer. We don't know how many enzymes, 1, 2, 3 of them, whether they are present or not, and what are the expression levels of this in cancer, and what's the ratio between them. Are this fixed ratio between the 3 enzymes or not? And is the expression correlated to the patient clinical features or not? I'm just moving to the logical framework, what we thought about this study. Then if we found that the 3 enzymes are present, so we try to knock down the enzymes to see the influence on the other enzymes. If we knocked 1 or 2, this influence the hydrogen sulfide level or not, and if it has an effect on the other actually hydrogen H2S synthesizing enzymes. And then we move to the third phase of the study, which is pan-targeting. If you find the 3 enzymes, can we pan-target or can we inhibit the 3 enzymes? Because it has been shown, and also from our earlier studies, that there is overexpression of hydrogen sulfide in breast cancer in general. So if we target the 3 enzymes, is it be quite useful to reduce the oncogenesis of the breast cancer? So this is the steps that we have done, and I'm going to go through the results of these studies. The first part of the study, we studied the expression profile of the 3 enzymes in Egyptian female breast cancer patients. We collected breast cancer tissue from 25 females with the normal extremities as a control, and the average age was 46.36 years. Actually, as you see in this figure, this is the expression level of the 3 enzymes using qPCR. You can find that as you compare the breast cancer tissue, this is the breast cancer tissue on the right, and on the left is the normal extremities from the same patients. You can find that the 3 enzymes are overexpressed in these patients. Of course, this is not all triple-negative breast cancer patients. They are all different types, but overall, these enzymes are overexpressed in the the breast cancer patients. So the next question, are the ratio between the 3 enzymes are constant or they are relatively fixed between the patients? It seems to be not. This figure shows actually each 3 columns beside each other represents a patient. You can find there are various proportions between the 3 enzymes in different patients. Most of the patients have CBS more elevated than the others, but you can see here there are quite variations. Some patients have 3-MST, the red one, higher than the 2 other enzymes. Some have the CPS higher than others, and as you see here, there are quite variations in the expression of the 3 enzymes in the breast cancer. For, of course, unknown reason, we don't know, but we don't have sense why this might be happening, but this is our finding. This is also reflected, of course, on the level of serum hydrogen sulfide, and we found that serum hydrogen sulfide is widely elevated in breast cancer patients compared to healthy controls. And when we correlated the level of the enzymes with the clinical features, we find that there is significant elevation of CBS at the left and CSE in the middle, as well as hydrogen sulfide, serum hydrogen sulfide in patients with high Ki-67, which is a proliferation index that reflects the prognostic marker for the breast cancer patient. So the more severe is the breast cancer, you can find more elevation of the enzymes of hydrogen sulfide. It seems that hydrogen sulfide is quite crucial for the growth of the tumor tissue. This is also reflected on 3-MST, the 3rd enzyme, which when we made the stratification of the patients based on 3-MST expression, we find that high 3-MST expression was correlated with younger age, pre-menopause, larger tumor size, and high expression of Ki-67. So it looks that the enzymes are well correlated to the clinical features of the patients. The more severe behavior of the tumors, the more overexpression of the enzymes. But when we looked or compared the triple-negative breast cancer with non-triple negative breast cancer, we find only CBS is significantly elevated in those patients compared to others. Only in CBS, not in the others.
Dr. Muhammad.
أيوة. مش هو أنا بادي 11 و10؟
يعني
طيب أنا عموما الربع ساعة اللي في الثاني مش هتبقى موجودة. فيعني I saved 15 minutes هنا. فأنا بس ده أنا لسه راوت 15 أنا ببص على الساعة حوالي ربع ساعة دلوقتي. فاضل 10 minutes. Then we moved to knocking down of the experiments. We looked at the impact of CBS knocking down on CSE expression in triple-negative breast cancer. We found here quite remarkable that when we knock down CBS, we find here, as you see, CSE is elevated. And this was surprising to us. It seems that there is a compensation mechanism to keep the hydrogen sulfide level high inside the cancer cell. When we knock down one, the other one is quite elevated. And this was on the messenger RNA level and also as well on the protein level, as you see here. When we knock down, at the last figure at the beginning, at the right here down, when we knock down both CBS and CSC, there is a remarkable decrease in the hydrogen sulfide production on these patients. For the third set of experiment, that when we found that the three enzymes were overexpressed, so we tried to pan-targeting the three enzymes by using microRNA, and then to go fast at this by using different platforms of bioinformatic analysis based on seed match, binding score, and binding energy. We found two microRNA that are effectively can inhibit, and here this can inhibit the three enzymes. One of them is 193, and this is the matching with the enzymes, and the second one was 548. And this is the matching without going through. First, we looked at the expression profile of these two microRNAs in breast cancer patients, and we find that both microRNAs were actually down-regulated in breast cancer. So then we tried to do a transfection experiments in MDA MB231, which is triple-negative breast cancer. We made a transfection of these two microRNAs, and this is the transfection efficiency that were overexpressed by this way. And then we looked at the impact of microRNA 193 on the H2S enzymes. As you see here, that these enzymes quite effective in suppressing the three enzymes here. And this is also similar for 548. Also, it was very effective in inhibition of the three enzymes. So they are very effective tool actually to inhibit the three markers or the three enzymes that synthesize hydrogen sulfide. And also, they were very effective in reducing the hydrogen sulfide level in those patients, which, as I mentioned to you earlier, hydrogen sulfide is very important for angiogenesis and for the tumor growth for breast cancer. So the final part of the experiment to see the impact of ectopic expression of microRNA on the triple-negative hallmark, which is the functional activities. And we find that when we transfect the cells with either 193 or 548 microRNA, as you see here, it reduced remarkably cellular viability compared to control. Other lines here are control cells. And also were effective in reducing clonogenicity or colony-forming assays. These are the quite functional activities for the breast cancer cells. Also, they're quite effective in reducing the colony-forming assay and also for the migration capacity. These are some indications that actually these microRNA are quite effective in the reduction of oncogenesis of the breast cancer cells. So reaching to the conclusion, hydrogen sulfide synthesizing enzyme, the three enzymes are overexpressed in breast cancer tissues. The 3-MST had higher transcript level in breast cancer at young age, premenopausal, high expression of Ki67, as well as CBS and CSC, and have larger tumor size. Both these microRNA 139 and 548 was really successful for they were underexpressed in breast cancer, but they are validated pan-inhibitors of the three hydrogen sulfide synthesizing enzymes in triple cancer cells and attenuate H2S. And this is a remarkable finding because it's really, we have been working with different inhibitors, actually pharmacological inhibitors, and there are controversial results about their impact on the functional activity of cells. And finally, microRNA 139 and 5 are novel tumor suppressors in breast cancer that act through simultaneous repression of CBS, CSC, and 3-MST enzymes. These are the conclusions. We have also other results on the immunological properties of cells, but because of limited time, I'm not going to show. I would finally like to thank my and colleagues or the group we have in the German University in Cairo. They are from pharmacology and biochemistry department. And this work has been funded by the Swiss National Foundation. And also like to thank my colleagues or collaborators in this project, Professor Chaba, which is very famous professor in the field of gasotransmitter. He's a star in the field of hydrogen sulfide, in particular from University of Freiburg, Switzerland, and also Professor Carol Balquin from University of Geneva for their contribution in this work. Thank you very much.
Thanks very much, Dr. Muhammad. Thank you.
So the next speaker will be Professor Muhammad Salim, Tanta University. And he gonna talk about the triggering toll-like receptor pathways, conditions, tumor reaction, CD8 plus T cells, better expansion in an adaptive transfer mouse model. Professor Muhammad, he's the founder, director of Center of Excellence in the Cancer Research, and the chairman of Egyptian Association of Cancer Research. Okay, please. We have 25 minutes.
Thanks. Just to give me a minute to... Sabah al-Khair, morning. Thanks so much for Professor Azazi for inviting me for this talk. And actually, at Tanta University, we established the Center of Excellence in Cancer Research. And in this center, we are interested in several projects. So one of these projects is about how TLR can modulate T cells in vitro and in vivo. This is what I'm going to talk about today. Other project at CCR includes cancer biology, the escape mechanism, either intrinsic or extrinsic mechanism. In case of cancer immunotherapy projects we are working on, we focus on vaccine-based therapy, especially dendritic cell-based therapy, and adoptive T-cell therapy. Today, I'm going to talk about sub-project of adoptive T-cell therapy. And just to give you background about both vaccine-based therapy and adoptive T-cell therapy, on the left, vaccine therapy, it can be done using peptide, proteins, RNA, DNA, either free or packaged on dendritic cells. We are interested in dendritic cells, generation of dendritic cells loaded with peptides. Vaccination can induce T cells to migrate to and traffic to tumor microenvironment to attack and destruct the tumor cells. On the other way around, adoptive cell therapy, we harvest T cells or immune cells from blood or tell tumor infiltrating lymphocytes, cultivate them in vitro, activate them, and then infuse them back to the same patient, or in case of animal study, with the hope they can traffic to tumor microenvironment and attack tumor cells. We focus on 3 phases of adoptive cell therapy, either during generation of T cells in vitro or after adoptive transfer of T cells. In case of generation of T cells, we use or focused in the last 10 years on 2 molecules. One is IL-12, and we, in a series of studies, about 12 studies, we found it's a magic cytokine which can activate T cells in vitro and induce their survival, trafficking molecules, and anti-tumor efficacy. Recently, we started to use TLR ligands which can bind to TLR expressed on
immune cells, and this is what I'm going to talk about today. We use also in combination with chemotherapy because we found the chemotherapy, especially cyclophosphamide, can induce beneficial microenvironment for the adaptively transferred T cells. And of course, we use peptide vaccination after adaptive transfer of T cells, and we again use TLR ligands. So I raise three questions, main question. How about TLR, can they really triggering this pathway in benefit cancer immunotherapy? Second one, how does triggering the TLR signaling pathway in vivo can impact on anti-tumor T cell response? And the third one, if we can modulate in vitro and in vivo. And I'll give you some, actually, question number 2 is based on, we answered already, or about 70% of this question based on our series of studies. The third one we started recently and we are working on. First, I'll give you some rationale about the link between TLR and immunotherapy, and we focus on immunotherapy because, as you know, all of you know, immunotherapy induce memory response. So it can, it induce sustained response against the tumor. And I enjoyed actually the talk of Dr. Ahmad Sultan today about salmonella because it's a kind of drug delivery which can induce inflammatory response which is required for anti-tumor response. If we look at this cartoon, tumor cells can be found and can be eradicated by what is called immunosurveillance. It's immune cells can attack on a daily basis or a monthly basis to destruct tumor cells in the best scenario. However, under certain conditions, this doesn't happen. And the tumor grow, and the tumor shed some tumor antigen, and the tumor antigen are baked up by dendritic cells. And then dendritic cells supposed to traffic to lymph node and present this tumor antigen to T cells, either CD4 and CD8, or both, I mean. However, this doesn't occur in tumor-bearing host because there is no danger signal. In case of infection, it's easy to get immune response. In case of because there is a foreign molecule. But in case of tumor, you don't have foreign molecule. You don't have danger signal. You don't have infection. So it's a self-antigen. So it's hard to get the response. That's why we are trying to provide something which can induce danger, mimic danger signal, and can induce inflammatory cytokines which can activate dendritic cells. In tumor microenvironment, if you look at under microscope and analyze by flow cytometry, immunohistochemistry, will find heterogeneous population of cells, including, of course, tumor and fibroblast and other cells. And you will find immune cells, but 90% of these cells are in a case of dysfunction. So we need to revive them in vitro and then transfuse back into the tumor microenvironment, either by vaccination or, in this case, adaptive cell transfer. So you will find different cells like Treg, MDSC, NK, and TAM2, type 2 macrophage. All these cells produce molecules which inhibit tumor microenvironment. So as immunologists, we need to have 5 signals to generate effector immunity in case of tumor. The 5 signals, signal 0 and the other 4 signals. Signal 0, it means danger signal. This can be provided by either infection, acute infection, and of course we don't, or microbial products which mimic the infection but sterile infection. And this includes PAMs, pathogen-associated molecular pattern, or DAMP, which is disassociated molecular pattern. Either this or this, you can get some activation of plethora of immune cells, most of subtypes of innate immune cells, and this can, of course, induce activation of dendritic cells. Then you can get signal 1, which is binding of TCR with antigen. Signal 2 is the costimulatory molecules. Signal 3 is the inflammatory cytokines. I mean signal 4, sorry. So you need to have 5 signals. One of the most important signal, or you can mimic signal 0 by TLR. Signal 0 and the signal, because signal 0 results in signal 4 or 5, which is inflammatory cytokines. TLR are toll-like receptors expressed on every cells, but they are highly expressed on innate immune cells, especially dendritic cells and the macrophage. Two types of TLR, some of them are expressed on cell membrane, others are expressed in endosome, and these are TLR which recognize nucleic acids, either single-stranded RNA, double-stranded RNA, or DNA. We focus in our studies on two of them, two ligands of these, which are TLR3 and TLR9. When TLR ligand bind to TLR on dendritic cells, for example, you get signaling pathway which by the end of the day, it activates NF-kappa B. NF-kappa B is a master transcription factor controlling production of pro-inflammatory cytokines and inflammatory cytokines. In most cases, TLR, especially TLR3 and 4, of course, 7, 9 ligand, they can induce pro-inflammatory cytokines. These cytokines, of course, and this was expecting to activate dendritic cells, and in this case, dendritic cells can be professional and super professional antigen-presenting cells to present antigen to CD4 and CD8. And this is what we are looking for, when CD4 can provide help for CD8, and CD8 can kill tumor cells in presence of antigen, specific antigen. This was the rationale behind our thinking, and the question too, how about if we provide TLR ligand in vivo, in a tumor model? We used in transgenic B-male transgenic mouse model. In this mice, CD8 T cells are engineered to recognize peptide on melanoma, which is class 1 peptide. So we get spleen cells from donor, activate them in vitro, transfuse them into the recipient. Recipients are bearing a tumor, but they are not engineered. But only the cells which are donor cells are transferred are engineered. So in this very nice model, you can easily discriminate or characterize donor cells from recipient cells, because donor mice are live 5.1 allele and the recipient live 5.2 or opposite. So it's a very nice model and a clean model. You can gate on CD8, T cells, either CD4 or CD8, and then you can, but in our case, we use CD8, and you can characterize their phenotype, activation, function, and the trafficking. So in this first group of studies, we used BOLA-IC. BOLA-IC is synthetic double-stranded RNA, which mimics natural double-stranded RNA, and is recognized by TLR3 on innate immune cells. The mic is working? I don't know. But I think I'm okay, right? My voice is okay? So we use the BOLA-IC as adjuvant system in our adaptive transfer model system. So we transferred the cells and then vaccinated the mice with or without BOLA-IC, which is TLR3 ligand. And we found dramatic expansion of the cells, transferred cells, only with the BOLA-IC in spleen, draining lymph node, and peritoneal exudate cells, and even in liver. And this was associated with very effective anti-tumor immunity against melanoma and lymphoma, which is not, this is lymphoma and also melanoma. So it means provision of or triggering of TLR3 signaling pathway in tumor-bearing mice during vaccination can induce expansion of adaptive transferred cells, as well as induction of anti-tumor immunity. So we thought, how if we combine chemotherapy with TLR3 ligand? Chemotherapy, we use the cyclophosphamide because it's widely used, and we used here as a model of chemotherapy. So we inoculated mice with tumor, then treated mice with chemotherapy, and then adopted the transferred cells, and then vaccinated with or without BOLA-IC, which is TLR3 ligand. As you can see here, with peptide and Bull-IC, you can get, as expected, good response. However, when these mice were pre-treated with cyclophosphamide, low dose of cyclophosphamide, you get very high response than Bull-IC and peptide alone. So it means something happened in microenvironment when you treat mice with cyclophosphamide before vaccination and adoptive transfer. Of course, we have done a series of studies, about 15 studies, to dissect what is going on after in this microenvironment. And then we published in Cancer Immunology and Immunotherapy some years ago, this 2 phases occurs after the cyclophosphamide treatment. Lymphopenic phase, in which cells are endogenous cells die, cytokines, high level of cytokines is there, dendritic cells are activated. And at recovery phase, cells start to recover through mobilization of stem cells from bone marrow into peripheral blood. And we found massive expansion, and this was something breakthrough from our group. We found a massive expansion of immature dendritic cells in blood starting from day 7. And we were able to activate these cells with Bull-IC and induce them to traffic from blood to lymph node. And this was something very interesting. And we dissected, based on this dissection, we come into what is called the tripartite regimen for immunotherapy. In this regimen, we start with chemotherapy, and then adoptive cell therapy, and then vaccination with TLR3 ligand, in this case, Bull-IC, and then later on, we repeated the vaccination. Using this tripartite therapy, we were able to get survival, 100% survival of mice, as you can see in the upper line of the figure, as well as we got very low tumor size when you have this tripartite regimen, but not in other regimen. You can get initial response, but tumor recurrence occur. But here you get sustained tumor regression. And we found at the site of vaccination, we found, as you can see here, the circle around the tumor size, which of course, with the tripartite regimen, the right one, it's almost gone. And we found white here, which is indicate to the vitiligo, which is because immune cells attacked the melanocytes, which is okay in this case because it's melanoma. And when we injected cells, tumor cells, melanoma, in blood, they, of course, traffic to lung to form lung metastasis. And we applied the same tripartite regimen. We were able to get rid of all the tumor cells. And those really was very, very interesting finding. And the tumor didn't come back, at least in this situation, for 2 months. So we can, the conclusion from, or the mechanism behind the TLR in vivo, based on these studies, is that Bull-IC bind to TLR3 on dendritic cells, macrophage, NK cells, and we proved that. And these cells are activated immediately in few seconds to minutes, and then they release pro-inflammatory cytokines. And these cytokines, of course, they are increased even after low dose of treatment with cyclophosphamide and another wave of TLR. And in presence of peptide and vaccination, CD8 T cells adopted the transfer, not endogenous. No, we are talking about the transfer T cells. They show better expansion, better survival, better memory, and better anti-tumor immunity, as I showed you. And based on this series of studies, we run clinical studies on pancreatic cancer patients, but using the dendritic cells, and this was published a couple of years ago and discussed in ASCO, which is the hematology, American Association of Hematology in US. So the last question, how about if we trigger TLR in vitro and in vivo? And can this happen also with other TLR? So we used our system, which is already established for adaptive cell transfer, and we, in a series of studies, about 15 articles, we were able to show that IL-12 treatment of CD8 T cells in vitro for 3 days with IL-12 can induce better survival, better trafficking, better proliferation, better activation, better anti-tumor response of CD8 T cells in vitro and when they are transferred in vivo. And this is, of course, established by our group using a female mouse model. We use the same in this. I show you just 2 slides as representative slides of anti-tumor effect of CD8 T cells treated in vitro with IL-12. And this is the tumor growth, and we compared with other cytokines, IL-2, IL-7, IL-15, and as you can see here, with female IL-12, it's like a magic, really. It eradicates tumor, in this case was melanoma. And this is represented in one mouse. This is control, cells transferred but without any treatment in vitro, and the right one with IL-12. So we use this system to ask whether treatment of cells with Bol-IC, which is TLR3 ligand, or CBG, which is TLR9 ligand, can induce similar effects as IL-12. CBG is unmethylated DNA. It can usually in bacteria and the others. And we use the same model. We got the donor mice. We got spleen cells unfractionated in this model. We use the unfractionated, not purified T cells because we wanted to see what is going on in culture. And this is due to direct effect on T cells, or it's indirect through other cells. And we then transferred these cells in vivo, and we looked at the expansion. First, we looked at proliferation and cytokine production. We found CBG actually is higher than TLR and higher than IL-12, which was something attractive for us. Then we looked at, we labeled the cells with CFC, and when cells divide, they lose, or CFC is diluted. So when you have more dilution of CFC, it means more proliferation. So we found, sorry, it's here, the proliferation of B cells in the culture, not T cells. So when you have in the culture B cells are proliferating, and this is regardless of the presence of tumor, even tumor antigen in vitro. Either you add or not, B cells can respond. And this is expected because B cell express TLR39. But what is not expected is the massive proliferation of B cells. And when we looked at CD40, which is a typical activation marker and the maturation marker on B cells, we found high level of this molecule. And we looked at CD8 T cells on the right. When they downregulate expression of what is called CD62L, it means these cells are activated because this is a trafficking molecule. So both proliferation, CD40 expression on B cells, it confirms the stimulation of B cells by CBG. And of course TLR, but CBG was much higher than about 10-fold than Bol-IC. Then we looked at different activation marker on B cells, only on B cells, because we found this massive effect on B cells. We get it on B cells, and we looked at CD25, CD69, CD40, CD80, CD86, and with or without peptide, with or without CBG in this case. And we found again, especially CD25, CD86, which is another typical activation marker, they are expressed regardless of presence of tumor antigen. And then we asked if we transferred these cells treated with Bol-IC or CBG in vitro and into recipient mice, are they going to expand in vivo? And yes, they expand, especially after, and we compared with IL-12. And we found again CBG, which is TLR9 ligand, is much higher than TLR3. And then we asked the last question. If we treat the cells in vitro with TLR3 or 3 ligand or 3,9 ligand, and then transfer them and treat the mice with either of them or the opposite, TLR3 in vitro, TLR9 in vivo, TLR9 in vitro, TLR3 it's combinatorial based on this table. And we found the conclusion is that if we treat cells in vitro with TLR9 and in vivo with TLR3, this can induce the highest expansion of CD8 T cells and the anti-tumor response. I think I don't show here because this is in press right now. So the conclusion is that CBG is superior to poly-IC and IL-12, which is, for me, was something really dramatic, to condition T cells in vitro. The in vitro effect is mainly mediated by a bystander effect on B cells. In this case, I guess B cells act as antigen-presenting cells, or not necessary. In case of peptide, it can act as antigen-presenting cells, but as a bystander effect, and produce cytokines and help for CD8 T cell. The in vivo effect is mediated by dendritic cells, and the conclusion, CBG in vitro followed by poly-IC in vivo is the best combinatorial treatment for better T cell response and induce anti-tumor and induce memory response. And this is my team, and thanks for them, and thanks for my team or my collaborators at Medical University of South Carolina, and thank you very much.
Thank you, Professor Mohammed, for this interesting talk. And a couple of questions. I will talk to you in the break, so it's very nice talk. So the next speaker will be Professor Rabi'i Hassan, and he's gonna give us talk about the electrochemical nanosensors, the future of health diagnostic, Zuwayl University Science and Technology, 6 October, Egypt.
The slides are here. Which one? Assalamu alaikum. First, I would like to thank Professor Hassan Azizi for the invitation, and special thanks for the Egyptian Committee for Biochemistry and Nuclear Biology for organizing this event. And I'm very happy to be here to share with you and take you to short and fast journey showing you the importance of biosensors in diagnosis of special diseases. The work has been done entirely in the Zuwayl City of Science and Technology here in Cairo, either with national or international collaborations. And when we ask ourselves as researchers and scientists, what kind of problems do we face in Egypt, or what the challenges in the globe internationally, we all think about water scarcity or energy resources. And sometimes we think about money and low income. But we are not so far from COVID-19 pandemic and microbial infection diseases. And I think this is a very severe warning and alert for infection disease and viruses especially could affect our life. And we could stay at home for years and months. That's why we always say infection disease, we have to pay too much attention, and not only to pathogens like microorganisms and viruses and fungi, but for the effective treatment as medical doctors or researchers, We always say الوقاية خير من العلاج. Better or early diagnosis is very necessary and to provide effective treatment. And I will not compare biosensors with the classical microbiological assessment like counting the cells or plate reading or nuclear techniques. But at least if I can ask that we have the capability to provide a new technology, let's say a new technology, and this technology can provide us with a very rapid and sensitive and selective diagnosis and detection for specific targets like cancer biomarkers, for example, or viruses or bacteria in different matrices. What is a biosensors? I think we are very familiar with this device. Biosensors like glucose monitoring in blood level, or to monitor the glucose in blood levels. Very cheap device. It's some 100 Egyptian pounds. I think 300 to 400 Egyptian pounds. And small plastic chip. And plastic chip costs around, I think, 10 Egyptian pounds after the inflations. And what you need is just a very small drop of blood. Put the blood drop on the chip and wait just for 15 seconds. This is a very short time to analyze and give you a result. And within a very short time and very low cost, you can measure or analyze your glucose level for how many times per day. So if I can say that a biosensor is something applicable and could be tailored for a different type of diseases and biomarkers, we should look into how the biosensor work and what the components of the biosensors. Biosensors have three main components. First one is that we call it biosensing elements or biorecognition elements. Biosensing element is very necessary to select one specific target. As I mentioned in previous slide, glucose is measured in blood. Blood is very complex matrix. Blood has so many, analyte has so many components, right? But we selected only glucose, and we measured the concentration of the glucose because we have a specific biorecognition element, certain type of enzymes, for example, glucose oxidase, to analyze or to break down the glucose. So if we say that we are targeting any unknown compounds and we could define specific biorecognition elements, for example, RNA or antibody or even cells, bacteriophage in some cases, and these biorecognition elements could be immobilized on the plastic chips, then whatever the matrix is, you can analyze with high specificity and high sensitivity. The second component is the transducer, which is the plastic chip. Transducer in our case is, we call it electrochemical sensor chip. And there are some criteria to select your sensor chip. For example, high conductivity, biocompatibility, and other parameters should be considered. Last is the processing of the signals. You have the samples, you have the recognition elements, you have the chip. You need to translate your interactions, biochemical or chemical interactions, to visible signals, for example, or in my cases, I will translate all of these biochemical reactions to electrochemical signals. We have so many advances in biosensor. Now, you can just wear a smartwatch or wearable devices, and you can analyze not only one target and non-invasive. You don't need to take blood sample from your body, but you can just rely on microneedles inserted on the scan, and you can see the level of different analyte like cholesterol, glucose, and so others. When we try to establish or construct a biosensor setup for a new target, we always say there are some certain considerations and have to be considered. For example, how the signals are transferred. Usually you immobilize, for example, enzymes or antibody, and the enzymes is reacting with substrate, and this reactions is taking place on the outer side of the surface of the sensors, and how the signals are transferred from the whole enzyme immobilized or protein immobilized on the plastic chip to reach to the conducting area, which is a very small area. You have to consider about the mechanism of electron transfer. In some cases, we rely on direct electron transfer or indirect electron transfer. This is very important. Another consideration should be taken into account. It's sometimes you can face the problem of false positive results. We call it non-specific binding. If you just leave out space on the chap, for example, just empty spaces, doesn't covered with the sensing element, then the binding of foreigner or interferences molecules could cause increase in the signals, which means you are not measuring the accuracy of the measurement is decreased because you have some other foreigner substances with the measurement. Last step for biosensor devising, you have to make sure that your biorecognition element, for example, antibody or enzymes, are well oriented and correct. The orientation is correct to face the physical binding with the substrate or the antigens. So the non-specific binding, the electron transfer, and the orientation and alignment of the biorecognition elements are very crucial and have to be considered when you start to think about construction of a new biosensors. In our research labs in Swell City, we just say we have a research lab for biosensors and we can do some specific tasks. For example, because we believe that nanomaterials play a very important role in biosensors, so we have a special unit for nanofabrications and characterization. Second, if you just worry about your analyte and you want to decide which substance, which biorecognition elements is necessary, we can also help you to design a very specific recognition elements. Third one is the applications, to apply the approach, biosensing approach for different diagnosis, for different applications, biomedical or environmental applications. In a very short time, I'm going to show you some applications we have in our lab. First, it's virus biosensors. And when we say virus biosensor, we always think about COVID-19. But actually, in Egypt, a special country, Egypt, Sudan, and Libya, we have a very dangerous animal virus, which is حمى القلعية, foot and mouth disease virus. And I think, I'm not a vet, but based on my group, they always say that about 30% of the infected animals, I think they have to be killed or they can be died, or at least you have to protect the non-infected animals from the farms. So 3-4 years ago, we had a project to fabricate electrochemical sensor for FMDV virus. FMDV, I think there are several serotypes for this virus. And in Egypt at the time, serotype Sep2 and serotype O were very common, and most of animal farms were, the animals were infected with these two serotypes. So I asked my student at the time, do we have a commercial antibody for the FMDV? They said, no, it's not available. So how to construct a selective biosensor for FMDV virus, and you don't have, for example, antibody, which is simple biorecognition elements. We directed our thinking to say, okay, we have a new technology called molecular imprinting technology, MIP. MIP could be used in case if you don't have natural biosensing elements. And MIP means you can imprint, real imprinting, your target within a polymeric matrix. And if you imprinted your target on a plastic chip and then you removed it from the plastic chip, you created a specific template, something like 3D fingerprint. And this 3D fingerprint with the small molecules or small particles like virus, I think they are not, they can create high sensitivity, but selectivity is not so high. We tried with this approach, and we published one in, I think in 2000, 2 years ago. We tested first the effect of nanomaterials on the sensitivity. And we said, okay, if we have a combination between gold, copper, nanoparticles, the sensitivity for imprinting of the FMDV will be increased. And after long validation and optimization for the sensors, we tested the calibration to see how far or to what extent we can measure the virus in samples. We have very high sensitivity, I think 0.1 nanogram per ml, which is very good detection limit. And when we try to test selectivity against different type of serotypes, the same virus, FMDV, but different type of serotypes. Actually, we have a very acceptable limit, but with some, if you combine or you have a mixture of viruses, I think you can have about 20 or 25% cross-reactivity. And we believe that if you are trying to avoid the natural biosensing elements, for sure you will face the cross-reactivity or interferences problems. But at least you can take a rapid decision. If you have, for example, 70% or 80% results showing that the animals or the patient is infected with this virus, you have to take a confirmatory step to make sure that, yeah, the virus and the serotype exist. As I mentioned in the beginning, we are not competing with the classical method, but at least we are going to provide a new technology. This can help you to take a very rapid and on-site decision to kill the animals, for example, or to protect yourself from the infections. So we have experience now to deal with the viruses, and when we face the problem with COVID-19 pandemic, we encouraged to apply for a project funded by the National Academy of Science here in Egypt. And we went to the virology lab in the National Research Centers, and we asked them to cooperate with us and to offer us the possibility to deal with the real viruses and try to imprint the virus. We thought about the imprinting from the first time because with just a few months after pandemic, so 3 months or 4 months after the pandemic, of course, there is no any company is producing the natural antibody for COVID-19. And we thought that, okay, that's really great. We have experience from the FMDV. We can apply the same technology for COVID-19. So we thought about nanomaterials. We selected tungsten oxide carbon nanotube as a basal, the basic component for the biosensors. And then we dealt with the real viruses. You can imagine just a few months after the pandemic, we have the real virus in our hand. Of course, in the virology lab, it's a 3 lab, so we take all the biosafety precautions. And just some optimization, like selection of nanomaterials. Here is the electrochemical techniques. One, we call it EIS, electrochemical impedance spectroscopy, is one of the very common electrochemical techniques. And the second one, left side, is the cyclic voltammetry, which is, we use usually these techniques to characterize and decide which nanomaterials are promising to be taken into the next step. So when we try to image the imprinted virus on the chip using the transmission electron microscope or AFM to make sure that we have an effective imprinting techniques and to go for further optimization. And this is the approach, as I said, imprinting for the virus particles and then try to see the sensitivity. You have very high sensitivity and you can measure the virus in real sample within 5 minutes. That's really great. But when we came to the sensitivity, the selectivity, we have a problem. We have a cross-reactivity problem. About 30%, I think, interfere with other coronaviruses. It's a very big family of viruses. So I said, okay, it's good that we don't have interference from, for example, human viruses or any other viruses, but the interferences came from this high similarity between the COVID-19 and other coronaviruses. So we confirmed our conclusion that MIB is good for high sensitivity, but not good for high selectivity. 1 year later, I think, we obtained the commercial antibodies for 2 different types of antibodies, and we immobilized these 2 antibodies on the chip just to avoid the cross-reactivity and interferences problems. And when we test the selectivity against different type of viruses, yes, we succeeded. We achieved the task, and we still say antibodies better than MIB in case if you are looking for high selectivity. But for the sensitivity, it doesn't matter. Both of them can provide high sensitivity. So I'm very grateful to my postdoc, Heba from Animal Health Institute, and I'm much appreciating the effort and cooperation with the NRC and with Canal 4, providing clinical samples and tools to deal with the viruses through this journey. Currently, we are extending our research to COVID-19, not to measure the viruses in clinical samples, but to measure the efficiency of vaccines through the antibody rapid test. So we will just inverse the biosensing approach. Instead of immobilizing the antibody, we will immobilize the protein and try to measure or detect the antibody to make sure that the vaccination process or the type of vaccines have an effective process. So second part from my lectures is to try to apply the biosensor for bacteria and toxins. And the application is not for biomedical actually, but for environmental and food quality monitoring. And we always try to find, let's say, better food or safe food. But not only food, and also water. And in some cases, water or food could be contaminated with bacteria or bacteria secreted exotoxins, which are very dangerous. So medical doctors have two options. One is to think about a very early diagnosis or better way for analysis. Or the second way, which we don't like it, is just to wait and see how the progression of infection disease is taken out to the patients and then try to find effective treatments. So we choose the best one, which is to provide very rapid diagnosis or detection of bacteria and toxins in some food, and very simple approach. It just to take samples from what kind the food is and put it on the chip and insert it to the device, wait for a short time, and then you will find the result. So the aim was to design these biosensors, and during 3-4 years ago, we established 4 different setups for bacteria. One for salmonella, and this is for minced feed, and we use the MIP, which is very cheap, and as said, in this case, we are not using antibody. Second approach is the antibody for Staph aureus in food as well, and both of them use the nanomaterials as a basic level for construction of the biosensor. Third one, which is cell viability assay, we are not using any kind of biorecognition elements, but relying on to measure the cell activity by electrochemical devices. Last approach was the bacteriophage, which is in cooperation with Dr. Ayman Shevin. He's here, and he will present his work afternoon. So these are 4 published approach, and you are welcome to read the details and the optimization and the result of them. But I'm going to show just a few slides. First one is the immunoassay for Staph aureus, and you can imagine that within few seconds, you can measure the concentration of Staph aureus if you have food contaminated with this kind of bacteria. And after immobilizing the anti-Staph aureus antibody on the nano chip, and just to try to, we always think about the selectivity, actually. It's a matter for us. So we found that either if you have a mixture or community of different type of bacteria, or you have Staph aureus alone, you have high sensitivity and high selectivity, and there is no any kind of interferences with from the other type of bacteria. Second one is the MIP, which is, I think everyone can think about MIP because you can save a lot of money, because antibody is very expensive and you have to wait for so long a time to just get it from different companies. So MIP, basically, we just took the suspension of Staph aureus, salmonella in our case, and put it with different type of polymeric matrices. And within a few minutes, you can prepare your chip, which is ready for detection of Staph aureus in meat or a different type of samples. Selectivity and sensitivity have been studied, and of course, the sensors can provide these very important advantages. Last class for the microbial detections, which we applied here for Candida albicans, with high direct electrochemical detections. In this case, we didn't use any kind of antibody or any sensing elements. But we just think about the electrochemical activity of candida albicans through its mitochondrial respiratory chain. It does redox reactions and electron transfer, and from this electron transfer, we can generate electrochemical signals. So from a mechanistic studies point of view, we detected that cytochrome oxidase, I think this is a terminal electron acceptor part in mitochondria of candida albicans, is regulating the direct electron transfer between the electrochemical chip and the living cells of candida albicans.
Dr. Rabia,
Yes.
2 minutes, please.
2 minutes, okay, thank you. Yeah, just to continue with the cell viability, we try to construct different type of approaches to measure the activity of cells, activity of bacteria, or activity of microbial cells. And we are not targeting to diagnose the cells, but we're supposed to say that if you have a potential drug target and the drug could affect the viability, why not we can use the chap to detect the concentration or the type of antibacterial agent or antimicrobial agent on the electrochemical signal. Then we can decide the sensitivity or resistivity of the microbes toward this kind of effective or potential targets. I think this is the last, because I have only 1 minute left. Biofilm. Biofilm, we always feel worry about bioinformation on biomedical devices and implant devices. And biofilm, if you have it, this means that you have to have open surgery and remove and decontaminate infections. That's why we have some studies, one to have real monitoring for bioinformation on metallic stents and see how the biofilm could form on the electrode to mimic the real state. And from the electrochemical measurement, we try to understand and give some recommendations to avoid or to prevent the bioinformation on different type of stents and catheters. For example, this one has been published just a few days ago to stop the bioinformation of eriginose and staph aureus on catheters and polymeric materials. So, biosensor market. In Egypt, we don't have real market for just to fabricate biosensors, and we are trying as Well City and Well City company to be involved in some market. Hopefully, we can find a space. Running projects in our lab, one for cancer diagnosis, breast cancer in specific, and we would try to provide one chip for simultaneous cancer biomarkers, not only one target, but maybe we can reach 3 to 5. It would be promising to say 8 biomarkers at the same time. The second one is to offer a technique for measuring the enterotoxin effect on cell lines and see how the cell, how the cell lines, for example, T cells, could respond to the enterotoxins that could be accessed in food samples, and how we could translate this interaction between toxins and immobilized viable cells to electrochemical signals. I have to appreciate, or I'm appreciating the support from cooperation between the Well City and national and international institute. My team, special thanks to everyone in my team for spending too much time in the lab, and for your kind attention and your invitation. And yeah, thank you so much.
Thank you, Professor Rabia. And okay. So we will start the coffee break.
Yes, I will just
yes.
Thank you very much for attending these five wonderful lectures. We are late for a coffee break, so we have the coffee break immediately now. It will be in Americana Plaza. Some staff in the back will guide you to the coffee break. Should be very short distance from now. Please be back on time at 12:30, because we have 4 exciting lectures. Professor Sami Ali, Hospital 57, and Dr. Ayman Shibini. We have Dr. Dua Gharib. She was unable to make it because she was ill. And we have Dr. Dalia Saleh from NRC replacing her. So we have 4 exciting lectures. Please be here on time at 12:30 sharp, please. I know, but I know, but we have be back at 12:30, please. 12:30. Thank you very much.